Hello, everyone. In this chapter, we are going to dive into the patterns of genetic inheritance. Now, as we dive into this topic, I would like you to focus on a number of questions. One is to ask yourself, what is a gene? Now, genes occur in multiple versions in the population. And these different versions of the same genes are called alleles. I would like you to ask the question, what is an allele? Some alleles are said to be dominant, others are said to be recessive. Ask yourself, what is this? What is a dominant allele? What is a recessive allele? What do we mean by genotype? And what do we mean by phenotype? Also, in order to predict the possible genotypes and phenotypes in the offspring, it is going to be important to draw what we call a Punnett square. I want you to focus on how to draw a Punnett square focusing on one trait and on two traits. Now, obviously, as you go from 1 to 2, the story becomes a little complex, right? Even more complex when we address 3 different traits. And what do we call a trait anyway? I want you to focus on that. I want you to ask yourself, what is a sex-linked gene? Or what do we call a sex-linked cross? What are some human genetic disorders that we typically study in genetics? Also, what do we call polygenic inheritance, multifactorial inheritance, sex-linked inheritance? Can we come up with some examples of sex-linked inheritance that are recessive, right? What do we call codominance and incomplete dominance? What is this, right? And oftentimes these days, many couples want to know what potential risk await for their future baby. So those who are doing implantation, right, artificial fertilization, are actually most of the time, not always, but sometimes do genetic analysis, genetic testing, right? And what kind of genetic testing is done before implantation of the fertilized embryo in a woman? So these are a few things I want you to keep in mind. I know these are a bunch of questions, but follow along with me. It's all going to make sense as we move on. All right. Our first step is to address the concept of genotype. What is a genotype? A genotype, it's basically your genetic makeup for a particular trait. For instance, your hair has a certain shape. Is it curly? Is it straight? This shape of your hair or the color of your hair is determined by your genetic makeup. In other words, what are the genes you receive from mom that account for the color and the shape of your hair? What are the genes you receive from dad that account for the color and the shape of your head, of your hair? I'm sorry. Sometimes there is a blend in things, right, that you receive from mom and dad. There is a blend. Sometimes one expresses clearly while the other one doesn't express, right? Sometimes the two express with an equal strength so that you actually see the two of them. We are going to take some examples. Now, whenever we talk about a genotype and we represent a genotype, we are going to have a representation of your genetic makeup as two different alleles. These two different alleles represent one you receive from mom and one you receive from dad for the very same gene, right? Now, the allele you receive from mom and the allele you receive from dad are always on the same chromosomes, right? Because the chromosomes are homologous, right? Remember when we talked about meiosis? We talked about homologous chromosomes, and the reason why they are homologous is because they don't only have the same shape and size, but also they have the same genes at the same location. Now, because those genes are not exactly the same in terms of sequence of DNA, there may be some small variations. These small variations account for our differences, right? So this variant of the same genes are called alleles. That's the definition of allele. I expect you to know that. I want you to know that. I will test you on that. Now, alleles always occur in two forms, right? For instance, here we can have, say, tall and short. Tall, capital T, representing the tall's height, right? And lowercase t representing the short height, right? I'm just saying anything right now, just for you to have an idea of what I'm talking about. So whenever we write capital T, that means that T is dominant. When we write lowercase t, that means that lowercase t is recessive. Now, what do we call dominant allele? A dominant allele, it's an allele that is going to be expressed systematically, is going to be expressed. Whether the recessive allele is there or not, it is going to be expressed. In fact, when a dominant allele expresses, it is masking the recessive allele when that recessive allele is present. So that in an individual with at least one dominant allele, what we actually see, it's the expression of that allele, right? So that this doesn't really matter and this doesn't really matter. All right? So that's a dominant allele. A recessive allele, it's a kind of allele or an alternate form of a gene that only expresses when the two forms of that recessive allele are present. For instance, an individual, for the case of short, right? That would be short. It would be lowercase t. Now, that lowercase t allows a phenotype short to be expressed. Now, obviously, in the case of human being, the story is much more complex than that, right? So don't really take it like literally, okay? I just want to give you an idea for you to comprehend what this is about. Let's move on. So, when we consider the genotype in an individual, the genetic makeup, now, that genetic makeup in an individual can be two alleles that are exactly the same. For instance, capital T, or capital A, In that case, because we have capital letters, these are dominant alleles. Because the individual has a dominant allele from mom and a dominant allele from dad, we say that this individual has a genotype that is homozygous dominant. Homozygous dominant. Homo means the same. Zygous means, well, the same cell or the same fertilized cell that carries the two dominant alleles. That's what it means. All right? Now, the individual can receive one recessive allele from mom and one recessive allele from dad, right? In that case, the individual is homozygous recessive, and that would be the genotype. All right? Okay? So now, what if the individual receives a dominant allele from mom and a recessive allele from dad, or vice versa? Right? In that case, for tall and short, the individual would be capital T, lowercase t, or capital A, lowercase a. Now, because these two alleles are not the same, we are going to call this particular genotype heterozygous. Okay? Hetero means a mixed, right? So one dominant and one recessive allele present in that individual for the particular feature we are considering. All right. So we've defined what a genotype is, which is a genetic makeup, right? What are the different alleles that are found in an individual, right? At this point, what we would like to ask is, now that we know the genotypes, what is the translation in terms of what we can see with our naked eye, what we can count, right? What we can observe, right? That would be the phenotype. So the phenotype is the physical or outward expression of the genotype. In other words, behind a phenotype, you have a genotype. Let me repeat that. Behind a phenotype, you have a genotype. You do not see a genotype, but you do see a phenotype. You cannot count a genotype, but you can count a phenotype. When I say counting, it's, for instance, you can measure the enzymatic activity of a particular enzyme that is produced because of gene expression. But let me close that parenthesis. I don't want to make things a little too complicated. All right? So we are going to ask this. If we focus on some particular features, for instance, when you consider your earlobes, are your earlobes attached or are they unattached, right? For attached earlobe, there is a particular genotype, and for unattached earlobe, there is a particular genotype. Now, for simplicity, for a long time, right, we've been teaching that an individual with unattached earlobe can be dominant, homozygous dominant, or heterozygous, because unattached is dominant over attached. That is recessive, homozygous recessive, right? That would be attached earlobe. But this is for very simplistic reasons that this is an oversimplification of what happens with attached, unattached earlobe. Now, here is the reality I want you to remember. I want you to remember that attached, unattached earlobe, it's not a clear cut. It's not basically two alleles that are occurring and that you have attached earlobe or unattached earlobe. The reality It's that further studies were developed and showed that attached and unattached earlobe, it's actually a continuous thread. In other words, it's not black or white. Okay? It is not black or white. That's a misconception. Some of the misconceptions that we have in human genetics. Now we know today that it's not a black and white thing or one thing or the other, right? There are many intermediate phenotypes. Anyway, let me close that parenthesis, but I wanted to make this clear because most of the textbooks you are going to see, if not all the textbooks, they teach that an individual who has unattached earlobe is either homozygous dominant or heterozygous, while an individual who is attached earlobes is homozygous recessive. That is not rigorously true. I want you to remember that. All right. So, before we move on, I want you to mark a pause. Ask yourself, what is your genotype? And what is your phenotype? What is a genotype? What is a phenotype? Look at your ears. Are your ears attached or unattached? Do you have freckles, or is it that you don't have freckles? Do you have a straight hairline, or do you have a widow's peak? Right? So these are things that you can kind of do to see, to check on you, some aspects of what we are covering here. All right, let's move on. Okay, so how do we come up with a genotype that ultimately is going to be converted into a phenotype? Well, we looked at meiosis, and we looked at specifically spermatogenesis, right, which is the formation of sperm cells, and oogenesis, which is the formation of egg cells. Now we have an egg cell, another egg cell here, another egg cell here. Now, remember that egg cells and sperm cells are haploid cells. In other words, for a particular trait that we are going to call A, right? That trait, we are going to say that it occurs in two forms, a dominant allele and a recessive allele. An egg can have a dominant allele. Another egg can have a recessive allele, right? So if a dominant allele egg receives a sperm cell that carries a dominant allele as well, this is going to produce an individual who is homozygous dominant, right? That individual who is homozygous dominant is going to have a normal pigmentation. Also, if an egg that has a dominant allele is fertilized by a sperm cell that has a recessive allele for pigmentation, we are going to have an individual, a zygote, that is heterozygous, right, with capital A lowercase a. Because capital A is dominant, normal pigmentation is dominant over no pigmentation, the individual is going to have a normal pigmentation. Now, what if an egg has a recessive allele for pigmentation and a sperm cell has a recessive allele for pigmentation? In that case, the zygote, after fertilization, is going to be homozygous recessive, which is going to have no pigmentation. In this case, we have an individual who is albino without pigmentation, right? Now, some genetic traits, for instance, short fingers versus long fingers, right? Freckles versus no freckles. Now, in humans, it appears that short fingers, it's actually dominant over long fingers. So that an individual with short fingers is capital S, or capital S, lowercase s, while an individual with long fingers is recessive homozygous, right? An individual with freckles is capital F, or capital F, lowercase f, while someone without freckles is recessive lowercase f. Now, how about crosses? What we call crosses, it's basically when you are bringing parents with particular phenotypes. Particular genotypes, right? You are going to consider individuals with different genotypes, so we're going to ask the following question. If you have an individual with freckle who is actually having a baby with an individual without freckles, what are we going to see? Or, not necessarily what are we going to see, what possibilities do we have in the offspring? All right? So we can determine, because of what we know about meiosis, we can determine the chances for having children with freckles and having children without freckles. We can say the same thing for widow's peak. We can say the same thing for the skin color, and so on and so forth, except that some features are controlled by one gene, some features are controlled by multiple genes, some features are controlled by genes and environmental factors. All of that needs to be taken into account. However, to simplify things, we are going to take a look at one trait cross, which is, for instance, that we are going to take a look at widow's peak, right? If you cross two individuals with widow's peak or straight hairline, I'm just saying anything, right? So we're going to consider an individual with the dominant, homozygous dominant genotype, and we cross that individual with another individual who is heterozygous. We are going to ask the following questions. Considering that one parent is homozygous dominant, the other parent is heterozygous, what are the chances that, right, we're going to have these genotypes, and what are the chances that we're going to have these phenotypes in the offspring? So we can do that with one trait. We can also ask the same question with two traits, all right? We can ask, for instance, okay, we are not focusing exclusively on individuals with widow's peak, but we are going to take a look at individual with widow's peak or straight hairline, and in this individual, do they have long fingers or short fingers? That would be two different features we are considering here, right? We know that in the very simplistic way, each of the feature is controlled by one gene. So that really what we are saying is that by doing a double cross or a two-trait cross, we are actually looking at two genes, right? And these genes occurring in different kind of alleles. Let me close that parenthesis. So now we are not just simply looking at homozygous dominant for W, but we're also looking at T, homozygous dominant T, right? And then we're looking at heterozygous W here and homozygous dominant T here. That would be a double cross. And we are going to ask the following question for whenever we do whether we are doing a mono cross or one thread cross or a two thread cross, we are going to ask the following questions. What are the gametes going to carry as alleles, right? Considering the genotype of the individual, say the individual is heterozygous, what can possibly be the gametes that are going to be produced from this individual? Now, when you have capital W and lowercase w, one is going to end up in one gamete, another one is going to end up in one gamete, so that really at the end of the day, whether you are talking about an egg or a sperm cell, they're going to have either capital W in a sperm cell or an egg, or lowercase w in a sperm cell or an egg. Well, this right here is a sperm cell. This right here is another sperm cell, right? Okay. So a parent who is heterozygous is going to produce two kind of gametes. One that carries capital W and another one that carries lowercase w. These are the two possibilities, right? Either capital W or lowercase w, but not both, right? Not both in a normal meiosis, right? Because if you find both, that means that you were in a case of a non-disjunction, right? Which actually would cause trisomy. We saw this when we looked at meiosis, right? Keep that in mind. But right now, we are not going to focus on dysfunctioning of meiosis. All right. So let's say that we are crossing two parents. One with no freckle, a male, and a female with no freckles. We know that freckles are, no freckles are recessive features, right? So the individuals are going to be homozygous recessive. So the sperm is going to have only one possibility, lowercase f. The egg is only going to have one possibility, lowercase f. And when you are crossing lowercase f and lowercase f, right, you're going to have a zygote or an individual that is lowercase f, so with no freckles. So in other words, there is no chance that two individuals without freckles are going to have a child with freckles. That is not going to happen, right? That's at least what this example shows. All right. So in terms of Punnett square, how do we come about predicting chances of having freckles or no freckles? Say we have two parents, a dad with freckles and a mom with freckles. Now, here is what we know about mom and dad. We know that dad is heterozygous, so capital F, lowercase f. Mom is heterozygous, capital F, lowercase f. Now, we are going to ask the following question. Mom is going to produce what kind of eggs? Well, some eggs are going to contain capital F. Some eggs are going to contain lowercase f. We are going to ask the same question for dad. Sperm cells are going to carry what kind of alleles? Capital F and lowercase f. Remember, mom is heterozygous, dad is heterozygous, right? So now, let's see what would happen if an egg cell get fertilized. Now, if this sperm cell fertilize this egg cell, we're going to have an individual that is homozygous dominant. So that individual is going to have freckles. If this sperm cell fertilizes this egg cell, the individual is going to be heterozygous with freckles, right? By the same token, if this sperm cell with lowercase f fertilizes this egg cell with capital F, the individual is going to be heterozygous, therefore is going to have freckles. While if this sperm cell fertilizes this egg cell, the individual is going to be homozygous recessive. Notice that you have, as we are crossing for one feature, as we are crossing two heterozygous individual, only one out of four is going to be homozygous recessive, therefore have no freckles. So now, with that said, so this right here, it's our Punnett square. So now, remember capital F produce freckles, lowercase f, no freckles, right? When the square is shaded like this, it's freckles. When the square is not shaded, it's no freckles. Now, in terms of phenotypes, we're going to have a phenotypic ratio that is 3:1. 3 individual with freckles, 1 individual without freckles. All right? So that's the phenotypic ratio. So in a mono-hybrid cross or a one-thread cross, you can be asked, what is the phenotypic ratio? How many individuals with freckles versus individual with no freckles do you have the chance to have in the offspring? That's the question, right? But another question can be asked. What are the different genotypes that are possible in the offsprings? Well, in terms of the genotypes, the story is going to be different because we have in reality 3 different genotypes. We have homozygous dominant, that's one genotype. Homozygous recessive, that's another genotype. And you have one of each, right? However, out of 4, 2 genotypes are heterozygous. All right? So that in terms of genotype, we're going to have a proportion or a ratio that we call 1:1. That's the genotype. Genotypic ratio. It's 1:1. 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. But the phenotypic ratio is going to be 3:1, right? All right, let's move on. Okay, so let's practice the Punnett square. So we are crossing a man with a genotype that is heterozygous, right? Capital F, lowercase f, with a female who has a genotype that is also heterozygous, right? Capital F is responsible for the presence of freckles, lowercase f, no freckles, right? So we have dad and we have mom. Mom would produce what kind of eggs? Eggs with capital F and eggs with lowercase f. Dad would produce what kind of sperms? Sperms with capital F and sperms with lowercase f, right? And now we are going to cross them. So we're going to cross capital F and capital F so that we have this genotype here. Then we're going to do the same thing here. Let me change my color. for clarity. Oops, I'm sorry. Let me change my color here. Okay, so now we are going to cross the sperm with this egg so that we have this genotype, right? We're going to do the same thing. This sperm and this egg, we have this genotype. This sperm here and this egg here, we have this genotype, right? This is how we do a Punnett square. All right, let's move on. So I mentioned the genotypic ratio. That's the number of offsprings with the same genotype. A phenotypic ratio. This is the number of offspring with the same phenotype, right? Observable traits, right? Okay. So in terms of the ratio, I already told you this. In terms of genotype, we have 1, 2, 1. Okay. 1, 2, 1. That's a genotype. Now, in terms of phenotype, we have 3 to 1, right? 3. Let me change my color here. 3. So 3 dominant phenotype, 1 recessive phenotype. All right? So we got that. So this is typically the example of what we call a monohybrid cross, right? Monohybrid cross. So for the freckles, we did that. And here, we can say the same thing for heterozygous parents, right? Capital A lowercase a and capital A lowercase a, we are bringing them together. We're going to have the exact ratio we mentioned earlier in the previous slide in terms of genotype and phenotype, right? But question though, what if we cross parents with freckles but homozygous dominant and parent with no freckles, homozygous recessive? Okay? In that case, all individuals in the offsprings are going to express freckles. Why? Because the eggs can only be lowercase f and the sperm cells can only be capital F, so that all individuals are going to be heterozygous and all individuals are going to have freckles. There is no other way around. Now, what if the parent with freckles is actually heterozygous? So dad would be heterozygous. In that case, dad is going to produce two kinds of sperm cells with capital F and sperm cells with lowercase f. Mom, having no freckles, can only produce what kind of egg, right? Lowercase f, right? So that 50% of individuals are going to be heterozygous with freckles, while 50% of individuals are going to be homozygous with no freckles, right? Okay. So a fair question we may ask is a dominant phenotype homozygous or heterozygous? To find out this, we actually do what we call a test cross. That test cross is going to cross an individual that we know to be homozygous and an individual that we don't know actually if they are heterozygous, or homozygous. But based on the outcome of the offsprings, if everyone is expressing the dominant trait in the offspring, then the parent who is dominant is actually homozygous dominant. While if we have in the offspring 50% of chance of expressing the dominant trait and 50% for the recessive trait, in which case the parent with unknown genotype would be a heterozygous. All right? That's what you need to know. All right. So now, this slide here shows you for two features, right? For two features, how is the distribution of chromosomes and how gametes are forming now. When we look at two traits, we're going to say that a chromosome that we're going to call set number 1 carries one of the traits. A chromosome set that we're going to call set number 2 is carrying another trait, right? Say, for instance, freckles and short fingers. All right? Short fingers is going to be carried by one set of homologous chromosomes, short fingers, long fingers, while freckles, no freckles, is going to be carried by another set of chromosomes, right? Now, during meiosis, how are the homologous chromosomes going to line up on a equator line is going to determine the genetic diversity, right? So, assuming that we are not taking into account crossing overs here, right? That's the assumption we're going to start with. We are going to say the following. We're going to say that, okay, well, individuals, in terms of possibilities of offsprings, based on the fact that we are only looking at two genes, we're going to have gametes that are capital F, capital S. Other gametes are going to be lowercase f, lowercase s. Other gametes are going to be lowercase f, capital S. Another set of gametes is going to be capital F, lowercase s. All right? So this is when we are determining the gamete outcome in an individual who is heterozygous, right? An individual who is heterozygous. So basically, that individual is capital F, lowercase f, capital S, lowercase s, right? So you're going to have, in terms of gametes, let me change my color here. Okay, so in terms of gamete, these two guys are going to end up in one cell. This guy and this guy can end up in one cell, okay? So that we have capital capital S and capital F, lowercase s. Let me change my color here. Then we can have, so these are two possibilities, right? And then other possibilities are for lowercase f now. So this lowercase f can end up in the same gamete as capital S. So F, capital S. Or this F can end up with this S right here, so that we're going to have lowercase f, lowercase s, right? 4 possibilities, okay? 1, 2, 3, 4 gametes possible. All right, let's move on. Okay, so in the D hybrid trait, considering what I explained earlier, now we are going to make things a little more complicated. Okay, so we're looking at two traits, right? So D hybrid cross. In the D hybrid cross, we're going to take a look at capital F, capital S, capital S, lowercase f, no freckles, lowercase s, long fingers, right? So The gametes here, there are only one possibilities for a dad with freckles and short fingers, right? It's going to be capital F, capital S. Likewise, mom here, right, with no freckles and long fingers, is going to produce eggs with lowercase f, lowercase s, so that the offspring are necessarily going to be short fingers and with freckles, okay? Short fingers with freckles. This is a D hybrid, all right? So that D hybrid is actually, if we cross that D hybrid, then we're going to ask what are the possible outcome for in terms of offsprings, okay? So this is when the 4 gametes that we produced earlier in the previous slide are going to come into play, and where we're going to see that the story is actually much more complex. So here we have our previous case, right? We have an individual who is double hybrid, but what if we cross a male and a female that have the exact same genotypes? Now, we are going to see that our gametes are going to be 4 kind for the female, 1, 2, 3, 4, but also 4 kind of gametes for dad, 1, 2, 3, 4. And we are going to cross each time the gametes, right? You cross this sperm cell with this egg cell, you obtain this. Then you cross this sperm cell with this egg cell, you obtain this, and so on. You obtain this, and here, and so on. So that all the possibilities altogether are 16 possibilities. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, right? But out of all these 15, the question we are going to ask is the following. What is the phenotypic ratio when we are crossing 2 individuals that are D hybrid? So right here. So capital F lowercase f, capital S lowercase s times capital F lowercase f and capital S lowercase s. What do we see? Well, what we see is that out of 16 individuals, 9 are going to have freckles with short fingers. 3 out of 16 are going to have freckles with long fingers. 3 out of 16 are going to have no freckles with short fingers. And 1 out of 16 is going to have no freckles with long fingers. All right? So this is the case of a double hybrid, right? Okay? So that's what we obtain. All right? Now, if we cross a double hybrid with a recessive individual who has no freckles with long fingers, well, in that case, we are going to have actually offsprings in terms of possibilities with, in terms of phenotypic ratio, 1 out of 4 that is going to have freckles with short fingers, 1 with freckles with long fingers, 1 with no freckles, short fingers, and 1 with no freckles, long fingers. Exactly the same ratio for each of the different phenotypes. All right? So that's basically how you can actually run a 2-thread test cross. All right? So again, in terms of the phenotypic ratio, If we are in a case of a monohybrid, right? Monohybrid, mom and dad, right? We're going to have a ratio 3:1 for the phenotype, right? Now, a monohybrid who is crossed with a recessive allele, it's going to be 1:1. In other words, 50-50, right? A dihybrid, double cross of dihybrid is going to be 9:3:1 for the phenotypic ratios, right? A dihybrid crossed with a recessive individual is going to be 1:1:1, right? Or 25:25:25:25. Altogether, it's 100, right? Okay. So that's what you have, okay? So let us stop our discussion here to resume our discussion in a next video. Thank you for your attention.